So we're going to talk about some more, just a little bit. We have not much time, short stint, about different aspects of mutations and mutagenesis and stuff like that. So what can happen, how mutations can be caused by chemicals or radiation. There are several types of chemicals that can cause mutations. The first pretty common type is nucleotide modifying agent, meaning that this agent, like nitrous acid, will change cytosine into the uracil, right? That makes sense. So cytosine used to pair with what? G. Now uracil is going to pair with adenine, so after next rounds of replication you have CG to AT substitution. Okay. And vice versa, A is going to be changed to hypoxanthine, which eventually will lead to the replacement of AT pair to GC. Now you can imagine that this is going to be a source of point mutations, right? Another type of chemical mutagens are intercalating agents, like this molecule called acridin. Intercalating agents. Now look at this. Look at this double helix over here. Intercalating agents get the molecules stuck, sort of incorporated between the adjacent nucleotide pairs, like my palm right now. That makes sense? So it's kind of shown on this picture over here. An acridin molecule is incorporated between the two adjacent pairs. You can imagine that if you try to stick something into the double helix, it's going to greatly distort the shape of the helix. Correct? That distortion will lead to deletions and insertions. I'm not going to ask you which strand which, but deletions and insertions eventually leading to frame shift mutations. That makes sense. Now, examples of these chemicals are acridin orange, like this one, and ethidium bromide. Now, acridin orange and ethidium bromide are reagents that are commonly used to identify DNA. Acridin orange is really useful to stain nuclei of the cells. You can basically add the solution of acridin orange to the cells, look down the microscope, and you can imagine that it will bind to the DNA, like it's exemplified in this picture. And all DNA in the cells will turn, not surprisingly, orange. Ethidium bromide is used to visualize DNA and the gel electrophoresis of DNA. Now, you don't need to know what they're used for, but they are extremely carcinogenic. As far as I remember, 1 or 2 drops of ethidium bromide applied to the mouse skin leads to skin cancer on the mouse. When I used to handle ethidium bromide in the lab, if I just need to move a vial with it, I would put on the gloves. I would never touch even a vial with my bare hands. It's one of the worst chemicals that you can work with. Now, examples of physical carcinogens are ionizing and non-ionizing radiation. Now, what happens in the ionizing radiation? X-rays or gamma rays have such a high power, high energy, that they will introduce single or double strand breaks. Does that make sense? A single strand break can be repaired, fairly easily, as you can imagine, because you still have a backup right here. So this break will distort the structure of the double helix, special enzymes will detect the distortion and they will remove the damaged part and will replace it with the proper. Okay. Double strand breaks much harder to fix. There is a whole system in the cell that is called double strand break repair system. Unfortunately, double strand break repair itself introduces mutations because you have mistakes in both strands. So anyone's guess. Does that make sense? Is that understood? There is a group of people, a limited group of people that are exposed professionally. It's a pretty cool profession. A lot of people dream of becoming those folks that are exposed to high energy rays, kind of X-rays, gamma rays, during their professional stints. That's really cool profession. Astronauts. Pilots, they slightly lower, so they still exposed, but exposure is lower. But when we talk about folks who are on the orbit, they're constantly exposed to high energy rays. And there's actually a whole NASA project on studying the double strand break repair mechanism to figure out how can we better protect astronauts from the potential mutagenic effects of space radiation. Why I know that? One person in the department where I worked, she worked on double strand break repair, and they had mice that were launched on the International Space Station to get them exposed. I think it was like enormously cool. I don't know how much it costs, but a lot. So now, ionizing radiation is terrible, but believe me, non-ionizing, which is UV light, may sound better, but it's not really better. So what happens in case of exposure to UV light? UV light can form so-called pyrimidine dimers. Most often those are dimers between two adjacent residues of thymine. You see, you have one thymine nitrogenous base and another thymine nitrogenous base. They form a dimer. This dimer greatly distorts the structure of the DNA. That distortion will lead to usually frame shift mutations during the subsequent DNA replication. Now, what do you need to do? What do you need to know about the pathogens? Mutagens, I'm sorry. So first of all, for nucleoside analogs, you don't need to know the examples. So what is nucleoside analog? It's the molecule that looks like a nucleoside, like adenine or guanine or thymine, but it's different. So if it is added to the cell, it will be inserted in place, like 2-aminopurine will be inserted in place of A. Will convert one pair to another and will lead to point mutations. What you need to know: nucleoside analogs cause point mutations. No examples, no mechanism for this. Am I clear? Number 2, nucleotide modifying agent. Nitrous oxide or nitrate. You don't need to know the nitrite actually. You don't need to know the example, but nucleotide modifying agent can cause point mutations. That makes sense. Like if I ask you, what kind of mutations can nucleotide modifying agent cause? Point. Clear? Intercalating agents. For this, I want you to understand what they do. At the level of, they stick themselves in between the pairs of nucleotides. Am I clear? And I want you to know they cause frame shift mutations. Yes. Intercalating agents, they basically stick themselves between the adjacent nucleotide pairs, distorting the structure of the double helix. And that will cause frameshift mutations. That good? No problem. Ionizing radiation. I want you to know that it introduces single and double stranded DNA breaks, leading to usually frameshift. And in some cases, ionizing radiation can lead to point mutations. I try to circle and highlight what you need to know exactly, what I'm going to ask. And finally, ultraviolet non-ionizing radiation leads to the formation of pyrimidine dimers, which then lead to frameshift or point mutations. Okay, now let me ask you this. Which of these chemicals and chemical and physical factors you get exposed? UV light, okay, when you tan. Okay, do you get exposed to X-ray? Sometimes, not often, sometimes, yeah. Gamma rays? Hope not. Are you exposed to nucleoside analogs? You aren't. Are you exposed to acridine orange ruthenium bromide? I mean, if you work in the lab and you really don't take precautions, then yeah, but you're exposed to this. That sounds really scary. Polycyclic aromatic hydrocarbons. Two options. First, you smoke. There are a lot of them in there. Second, you inhale directly from the tailpipe of 1975 Buick. Because modern cars have catalytic converters that burn most of that stuff to less dangerous chemicals. Okay. Now, what about nitrous oxide, bless you, or other derivatives of nitrogen, specifically nitrogen with a valency of 3? Depends on how much you like processed meat. Because, bless you, if you like it a lot, and it says cured, then you get exposed to a lot of nitrite. Okay, and nitrite is one of the derivatives that can do exactly this. Does that make sense? So, study that was published, I believe it was early 2018 or late 2017, that study came out in Britain. They did a large clinical observational study and concluded there is a strong association between the consumption of processed meat, essentially meat that was treated with nitrites to preserve color, and risk for developing colon cancer. Well, guess what? The study came out 2 months later. You walk into any grocery store and they have a whole bunch of uncured salami, uncured this, uncured that. Because, you know, the study got popularized a lot. You know, people started to ask, you know, can we just avoid nitrites? And food industry delivered, surprisingly. Okay? Or should I say not surprisingly. So we're going to talk about other aspects of the repair and all that crap next time. Just don't have time to properly talk about it. So until Thursday, see you then. I had a